Good morning, ladies and gentlemen. Thank you so much for inviting me to speak today. It's my pleasure to talk to you about the metabolomic signature of aniridia patients, which reveals systemic dysfunction. So some background. PAX6 is essential for the development of the eye, pancreas, and central nervous system. Haploinsufficiency of PAX6 is the underlying mechanism behind the panocular condition aniridia. And there is growing evidence for neuronal and endocrine abnormalities in patients. Some have been documented to suffer from obesity, hyperglycemia, insulin resistance, and diabetes. Others experience sleep disturbances, autism, ADHD, depression, and anxiety. We recently published a natural history study looking at the clinical features and disease progression of 86 patients with molecularly confirmed PAX6-related aniridia. When we delve deeper into the systemic features, we found that up to 13% of this cohort had type 2 diabetes, and this is twice the UK prevalence. Over 20% suffered from obesity, 7% had learning difficulties, and 2% had autism. So the aim of this study is to investigate the systemic effects of PAX6 through whole metabolomic profiling of genotyped aniridia patients. So we recruited 25 PAX6 confirmed aniridia patients and 25 age and gender match healthy controls. We looked at over 1,000 metabolites in the blood plasma of these patients, including proteins, carbohydrates, lipids, caffeine, etc. We asked each individual to fill out a food frequency questionnaire to ensure that the results were not skewed by their diet. And patients with WAGA, diabetes, or those taking statins were excluded from the study. So our cohort included 7 aniridia children and 18 adults. The mean age of both groups was 31 years, 48% were male, and the BMI of the aniridia group was slightly higher at 27.7 compared to the control group, which was 24. We had a range of mutations, 11 frame shifts, 10 nonsense variants, 1 splice, 1 whole gene deletion, and 2 patients with C-terminal extensions. The PCA plot showed little separation between the aniridia group and the healthy controls. But when we factored in age, we saw more of a differentiation. Several pathways were affected, and over 94 metabolites were shown to be statistically significantly different between the aniridia and healthy group. I'm going to go through 3 of the main disrupted pathways: oxidative stress, complex lipids, and serotonin levels. So we detected higher levels of oxidative stress in the blood plasma of our aniridia patients. The antioxidant taurine and its precursor hypertaurine were significantly reduced. Increased metabolites upstream of the glutathione synthesis pathway were found: cysteine, alpha-ketobutyrate, and cysteine. So, should we be considering increasing our dietary antioxidant intake? And should aniridia patients in the future be given high-dose antioxidant drug treatments such as N-acetylcysteine? We found disrupted complex lipids. Plasmalogens are a unique class of phospholipids with antioxidant properties and enriched in our polyunsaturated fatty acids. Ethanolamine-derived plasmalogens are mostly concentrated in myelin, which is highly abundant in our central nervous system. This particular group of plasmalogens and lysoplasmalogens are significantly reduced in aniridia patients. And so we need to further investigate this to understand the significance in aniridia. We found that there was an accumulation of ketone bodies despite no changes in glucose levels in our aniridia patients, so high levels of acetoacetate and beta-hydroxybutyrate. Other raised markers linked to ketoacidosis included methylsuccinate and isovalerate. Ketogenesis is mostly regulated by insulin, suggesting an altered insulin state in aniridia patients. Serotonin is a neurotransmitter produced from tryptophan in the pineal gland, and it requires PAX6 for its development. Low serotonin levels are mostly associated with depression and anxiety. And we found a 30% reduction of serotonin levels in our aniridia patients. Now, this hormone is also necessary for the regulation of sleep, mood, and behavior. So to conclude, this is the first comprehensive metabolomic analysis of aniridia patients with molecularly confirmed PAX6 haploinsufficiency. We found biomarkers linked to insulin resistance, possibly predisposing our aniridia patients to a higher risk of developing diabetes. Higher levels of oxidative stress may have cardiovascular implications, and low serotonin is linked to mood, sleep, and behavioral issues. We now need larger studies for verification of these findings, and we need to consider the holistic management of our aniridia patients going forwards. So with that, I would like to thank you and acknowledge my team, and in particular, Dr. Dulce Kunner and Dr. Vivian Kitt, who were focused on the Metabolomics project. I'd like to thank our funders, and in particular, the MRC, Aniridia Network, Sentinel Oncology, Moorfields Eye Charity, and the Wellcome Trust. Thank you very much.
London's Institute of Ophthalmology. The Institute of Ophthalmology was founded in 1948, originally based in Judd Street, London, and then moved to its current site at Bath Street in 1980, adjacent to our partner Moorfields Eye Hospital. We have a strong eye health research history and house around 200 staff, including 45 principal investigators. We have a range of facilities, including 2 lecture theatres, where we host our world-leading education programs relating to ophthalmology and vision research. We were awarded the best place to study ophthalmology in the world in the 2017 Centre for World Rankings. We have a specialist ophthalmic library in the basement where we provide students and staff access to all the latest scientific and medical journals. There is a small museum devoted to the history of ophthalmology, curated by Richard Keeler, where we exhibit the original surgical tools and instruments we have developed to aid our clinical practice over the years. From the ground floor to the 4th floor, we have corridor after corridor of research laboratories looking for the answers to a multitude of research questions. Welcome to one of the research labs. This is a typical example of what a basic science laboratory looks like. Lots of chemicals, molecular biology machines, and a hive of activity for our scientists. Several of the groups at the Institute are working on aniridia-related research. This ranges from trying to identify and understand the genetic changes in the PAX6 gene, developing disease models of aniridia, for example using human cells. We can now take a skin biopsy from a patient, convert the skin cells into stem cells, and then reprogram this to grow into an early eye cup with tissue that becomes a retina or a cornea. This really helps us understand how the PAX6 gene affects the eye tissues and allows us to develop and test new potential treatments for patients. We also have an accredited stem cell transplantation laboratory, so we can use tissue engineering to deliver cells directly to a patient's cornea. The Institute of Ophthalmology has a mission to promote and expand the unique eye research and education that we provide, and to take that from our labs to patients and the rest of society. In 2007, the first UK National Institute of Health Research Biomedical Research Centre for Vision was awarded to the Moorfields and Institutes Partnership. It remains the only biomedical research centre of 20 nationally awarded that is dedicated to a single specialty, with an outstanding track record for medical and translational research. Our close partnership with Moorfields Eye Hospital means that we can realise the vision of from bench to bedside in a seamless way, where the research is also aimed at clinical applications and where clinical requirements shape the research.